Hi, I'm Kelly Frazer, and I'm a professor in the Department of Pediatrics here at UC San Diego, as well as director of the Institute for Genomic Medicine. And the goal of my research is to try to understand how the genome works, specifically how genetic variants, these base pairs that vary between individuals, which of those variants are functional and how they function at a molecular level.
Cool. And then this particular study that is being published in Nature Communications, can you speak a little bit about that?
Sure. So what was known in the field is that genetic variants in regulatory elements are associated with differential expression of genes. So genes in different people have different expression levels, and this is related to genetic variants. And it was also known that the genetic variants, these regulatory genetic variants, are enriched in what's known as anchors at loops. So these high C loops. But it wasn't known exactly how the regulatory variants that are enriched in the anchors mechanistically work, i.e. how do they actually regulate the expression levels of genes. And we had thought that they would probably result in the formation or the abolishing of these loops. But what we actually found was that the loops are present, and what they do is they result in kind of subtle changes in the frequency between the contact of the loop anchor and the promoter, or the gene itself. So it's associated with this differential contact frequency.
Very cool. So to me, that sounds a little bit like a quantitative measure versus a qualitative measure. Is that a fair assessment?
Yes. So people before were thinking that the loops would either be present or not present. And what we found is that they're always present between different cell types, between different genetic backgrounds, haplotypes, but their frequency, that their distance as measured by contact propensity is slightly different.
And can you briefly touch on some of the technologies that enabled you guys to uncover these answers?
Sure. So what we did is we had a 3-generational family of 7 individuals, and we looked at 2 cell types from them, both induced pluripotent stem cells, iPSCs, as well as derived cardiomyocytes. And so we had 2 different cell types, as well as 3 generations of individuals, so we were able to look at to see how the genetic background influences this. So, and then we used the molecular data we had was the high C data, was generated using the Arima method.
Mm-hmm.
And we also had RNA-seq data from these cells, as well as H3K27 acetylation data and other types of molecular data sets. And so this whole combination allowed us to observe what's going on in how the quantitative changes between genetic variants that are associated with high expression of a gene versus genetic variants that are associated with lower expression of the gene, the 2 alleles of a variant.
Yeah, that's great. And then going forward, what are your plans taking this work to another level?
Well, so now we are incorporating it in a variety of our other studies. We are now looking to see how ataxic chromosome-wide levels, how there's co-accessibility in the ataxic peaks, and how does that interact or supported by these long-range looping mechanisms. We're also using it to, further help us pinpoint what genetic variants are likely to be functional in different cell backgrounds.
That's great stuff. Dr. Frazer, thank you so much.
Thank you.
Congratulations.
All right, thank you.